Hello, everyone. So first, special thanks to the organizers for this fantastic opportunity for us to share the recent work that we have done in this area. Let me share my screen. So can you guys see this really well? My name is Stephen Yi, Director of Bioinformatics at the University of Texas at Austin. So today I'm going to talk about emerging gain of function mutations in disease and their computational interpretation and characterization. So let's first start with this general question. So we have found in the past decades also, this is in the whole community, that there is a huge genetic heterogeneity in human cancer, as documented in a series of review articles and research discoveries. So as you can see here, so there's an increasing number of mutations identified in the past 2 decades also. When you look at different cancer types, for example, in this over 30 cancer types, different cancer types documented in TCGA, you see a large variation. This is for the number of coding mutations per sample identified in cancer patients. You see a huge variation in terms of mutation burden across different cancer types. So this leads to a fundamental question about genotype-phenotype relationships in cancer, as we are interested as a whole community to interpret different genotypes in order to review their functions and link them to phenotypes in health and disease. So apparently there is a black box. In order to uncover this black box and to make sense of the large number of genotypes or mutations, we are proposing a network-based approach, so-called network type. So put it in a simple way, there are 2 different scenarios. One is loss of interactions caused by mutation 1 here, and the other will be a gain of interactions, as you see here in the second mutation. In this scenario, it gains an additional interaction compared to the wild type network. So essentially, this deregulated gene regulatory network will be what we consider underlying different diseases. So we wrote a series of reviews in the past 2 or 3 years documenting this, and we have a very interesting term called functional variomics to indicate the field of large number of mutations and their effect in network perturbation. As you can see here, we illustrated in a graph different scenarios representing classic examples of effects of mutations. In the very top scenario 1, you have a loss of all interactions, typically is caused by a nonsense mutation, loss of function effect for this mutation. Or you have a JADIC, this is a term that came out recently in the past decades also. This case is like you lose a specific interaction compared to the wild type interaction network. It can also lose different types of different interactions. It can lose these 2 interactions or keeping the other 2 interactions intact, as you see here in this scenario. It can also be no effect on this network. So like in this case, the mutation 5 is a wild type scenario, is similar as the wild type network, or it can also gain an additional interaction compared to the network, as we mentioned just now. So I want to start with this 1,000 disease project that was done about 5-6 years ago, as what we call a landmark study, studying 3,000 missense mutations found in ISMB and 1,000 genome, the common variants found in healthy people. So we cloned them in gateway vectors express proteins, and then did a whole bunch of different characterization to study chaperone binding, protein interactions, protein DNA interactions, and post-translational modification and kinase and phosphorylation events. At the end of the day, you can integrate all of these different data sets documenting different types of molecular data to represent the effect of mutations in an integrated network fashion. So we can get a holistic view of network-based mutational effect for specific mutations found in disease or found in healthy people, like common variants. And this is very interesting, though we demonstrated by this study that functional variomics is an approach that we took in the past, in the last slide that I showed you, can distinguish different disease variants. So essentially you can stratify disease risk. So as you show here, by statistic, we found that roughly 30% of these disease mutations can be a cause and null, so-called phenotype. So this is losing all interactions. Another 30% also, these disease mutations, they cause an interesting genetic perturbation event, losing some part of the interaction, but keeping some other interactions or pathway signalings intact in the cell. Well, the other 40% are more like wild type. And this is verified later on by a very nice comparison using 1,000 genome. So common variants found in healthy people. You can see where you see 90%, the vast majority of these common variants found in healthy people, they are not changing protein interactions. So they behave like wild type. So this whole set of data validated our approach as functional variomics as a valid platform to study disease, to stratify disease risk, interpret different disease mutations. So we developed further a computational tool called Immuno. It came out earlier this year in NAR to predict potential effect of mutations in a cancer network. So we started with gene expression, protein interactions, and somatic mutation found in cancer. We compared the genetic or edge-specific profile with mutation-specific profile in each patient. And by a Monte Carlo simulation calculation, we were able to identify mutations that cause a specific change as an outlier in the whole population in order to identify a change caused by a specific mutation. In this manner, we were also able to integrate with yeast genetic information, genetic interaction data to identify pairs of mutations or pairs of genes that could be synthetic lethal as a prediction in cancer, the specific context. We went further to apply this tool to study cancer immunome in order to identify potential potential targets for immunotherapy. So again, we integrated mutation data. We focus on the 33 cancer types. We integrated mutation, gene expression, clinical features. And by that, we built a network model based on random walk with restart specific algorithm. We were able to compare that with existing drug gene signature based on CCLE cancer cell line data in order to identify drugs that could resensitize these resistant population of patients that are resistant to current immunotherapy treatment, in the hope that we can identify these new drugs that could help them sensitize to current immunotherapy. So we're able to identify a whole list of very interesting targets. So take-home message, network is really interesting to stratify genotype, phenotype relationships. We were able to identify different phenotypes and apply this into a cancer immunome context, and then identify gain of function drivers. I would like to thank all the collaborators and people in the lab, and we have positions available. Thank you very much.